Hey everyone, this is my first video on this channel of Biochemistry Tutor, and I really hope you will enjoy learning biochemistry through my channel. So today I'm going to talk about nucleic acids, nucleosides, and nucleotides. So nucleic acids, which you have already heard about, there are basically two types of nucleic acids. One is called deoxyribonucleic acid, that is DNA, and second is ribonucleic acid, which is called RNA. So DNA is a double-stranded molecule which forms an alpha helix, whereas RNA is a single-stranded molecule. Now, DNA is mostly located in the nucleus, most importantly in eukaryotes, and RNA is located outside the nucleus. Now, DNA is important in storing genetic information, and it's kind of like it acts like a cellular library, whereas RNA is important for transferring genetic information and as well as in protein synthesis. So basically, the genetic information flows from DNA to RNA to proteins. That means DNA is transcribed into RNA, and then RNA is eventually translated into protein synthesis. So both DNA and RNA are polymers, and polymers are nothing but they are basically just a long repeating units of monomers. And in this structure, these monomers are carbon, hydrogen, and oxygen atoms. Now, when it comes to both DNA and RNA, these monomers are called nucleotides. And nucleotide, which makes up DNA, is called deoxynucleotide, which contains a deoxyribose sugar molecule, which is a pentose ring, a 5-carbon ring, and it also contains a nitrogenous base which is attached to carbon 1 position of the pentose ring, and it also contains a phosphate group which is attached to carbon 5 position of the pentose ring, which is located outside the pentose ring. Now the nucleotide which makes up RNA is called ribonucleotide, and it contains a ribose sugar which is again a pentose ring, and it also contains a nitrogenous base which is attached to carbon 1 position and a phosphate ring which is attached to carbon 5 position of the sugar molecule. So the difference between these two sugar molecules, that is ribose and deoxyribose, is ribose sugar has 2 hydroxyl group which is attached to carbon 2 and carbon 3 position of the pentose ring, whereas deoxyribose has only 1 hydroxyl group which is missing a hydroxyl group at carbon 2 position, and therefore it is called deoxyribose because it is missing a hydroxyl group here on carbon 2 position. So basically, nucleotides contain a sugar molecule, a nitrogenous base, and a phosphate group. So now let's look at more detail about a nitrogenous base. So there are basically two types of nitrogenous base. One is called purines, which is a double ring. One is 6-carbon ring and another is 5-carbon ring. Now the second nitrogenous base is called pyrimidines, and which is only a 6-carbon ring, so therefore it is a single ring. Now the way to remember whether purine has a double ring or pyrimidine has a single ring, there is one way to remember, is because purine, it has a shorter name but it has a larger structure. At the same time, with pyrimidines, it has a larger name but it has a shorter structure. So this is kind of very nice way to remember, one way to remember that purine has a shorter name but has a larger structure because it has a 6-carbon ring and a 5-carbon ring, whereas pyrimidine has a larger name and the shorter structure. Now, the other thing about purines and pyrimidines are the 6 atom ring or the 6 carbon ring in purines are numbered anti-clockwise manner, whereas 6 carbon ring in pyrimidines are numbered clockwise manner. Now, purines includes adenine and guanine, whereas pyrimidines are thymine, uracil, and cytosine. So DNA contains adenine, guanine, cytosine, and thymine, whereas RNA contains adenine, guanine, cytosine, and uracil. So the difference between two is only DNA has a thymine, whereas RNA has a uracil. So that is the important difference which everyone, I guess, needs to remember. So now let's talk about a little bit in detail about purines and pyrimidines. So purines, like I suggest, it has a shorter name and a larger structure. So the basic structure of purines, as you can see here, it has one 6-membered carbon ring and second is 5-membered carbon ring. So if you remember, the six carbon ring of purine is numbered anti-clockwise manner. So let's number this ring. So on first position we have a nitrogen group attached, and there is also another second nitrogen group, nitrogen atom, which is on third position. So that means on the 6-membered carbon ring there are 2 nitrogen atoms and 4 carbon atoms. Now on the 5-carbon ring, the 5-carbon ring also has 2 nitrogen atoms, which is at position 7 and position 9. So here there is a hydrogen atom attached to nitrogen, and where there is no hydrogen, that means there is a lone pair electron of nitrogen. Now it also has a double bond between position 2 and 3, position 4 and 5, and position 7 and 8. So this is the basic structure of purines. So this is guanine, which is also the same basic structure of purines. Now let's look at the difference between adenine and guanine. So the one difference is there is an additional double bond in adenine between position 1 and carbon 6. Now there is a NH2 group which is attached to carbon 6 position of adenine, whereas in guanine there is a NH2 group which is attached to carbon 2 position of 6-carbon ring in guanine. And another important difference in guanine is there is a carbonyl group which is attached to carbon 6 position of 6-member ring. So this is the structure of adenine and guanine. So now when the sugar molecule is attached to adenine or suppose guanine, it is important to remember that sugar molecule can be only attached to position 9 of the 5-membered ring, which is called N9 position. That is the 9th position of nitrogen atom in purine ring. So this is very important, guys. You need to remember that the sugar molecule can be only attached on N9 position in purine ring. So then the important thing is when the sugar molecule is attached to adenine, it is called adenosine. Similarly, when the sugar molecule is attached to a guanine at N9 position of nitrogen atom, it is called guanosine. And both these attachment of sugar to adenine or guanine are called nucleosides. So nucleosides, basically they are just a sugar molecule which is attached to nitrogenous base. So this is nitrogenous base, and then when you add another sugar molecule, it becomes nucleoside. And most importantly, I'm emphasizing again that it is sugar molecule is attached at N9 position of the purine ring. Now lastly, when the phosphate group is attached to the sugar molecule, it becomes adenylate, and it is and in this structure, like I showed before, that the combination of a phosphate group and a sugar molecule and a nitrogenous base is called nucleotide. So again, when the phosphate group is attached to nucleoside, it becomes nucleotide. So here in this case it is called adenylate, but alternatively you could also call, like you can also name this nucleoside and which can be called adenosine, because this is adenosine. This is the basic structure of adenosine, and the phosphate group is attached to carbon 5 position of the sugar molecule, therefore it is called 5' monophosphate. So therefore it is called adenosine 5 monophosphate, that is AMP. And if there is two phosphate group attached to the sugar, it will be called adenosine diphosphate. If there are 3 phosphate, it will be called adenosine triphosphate, that means ADP or ATP. Now similarly, when the phosphate group is attached to guanosine, it is called guanylate, and alternatively it is also called guanosine 5 monophosphate. So again, just to clarify, this is our nitrogenous base, and when the sugar molecule is attached to nitrogenous base, it becomes nucleoside, and when the phosphate group is attached to nucleoside, it eventually becomes nucleotide. So this is very important, guys, to remember. Now let's look into pyrimidines. So like I said, pyrimidine has a longer name but a shorter structure. So pyrimidine contains 6 carbon ring, and let's look into the basic structure of all 3 pyrimidines. Those are thymine, cytosine, and uracil. So if you remember, the 6-member ring of pyrimidine is numbered clockwise manner. So let's number this ring. So this is the carbon 1 position where nitrogen atom is attached, carbon 2, and there is another nitrogen atom at carbon 3 position, and 4, 5, 6. So basically there are 2 nitrogen atoms and 4 carbon atoms in pyrimidine ring. And there is a double bond between carbon 5 and carbon 6. So this is the basic structure: 2 nitrogen atoms and 1 double bond between carbon 5 and 6. So now let's look at the difference between these 3, thymine, cytosine, and uracil. So the one common group which is attached to carbon 2 position is the carbonyl group. So carbonyl group is attached to carbon 2 position in all 3 pyrimidines, that is thymine, cytosine, and uracil. Now the difference is there is a hydrogen group, hydrogen atom on thymine and uracil on position 3. And the other difference is there is a double bond between position 3 and 4 of cytosine. And most importantly, there is a carbonyl group on thymine and uracil on position 4. So these both thymine and uracil, they look very similar. At this point they are very similar. The only difference is that there is a methyl group attached to carbon 5 on thymine, whereas there is no methyl group on uracil in carbon 5 position. So this is the only difference between thymine and uracil. And with cytosine there is a NH2 group which is attached to carbon 4 position. Again, this is not very important to remember the structure of purines or pyrimidines, so you don't really need to follow this, but most important thing to follow is the transformation of nitrogen base to nucleoside to nucleotide. So let's see how nitrogen base is now transformed into nucleoside. So like we saw before, that nucleosides are basically attachment of sugar molecule to the nitrogenous base. So when the sugar molecule is attached to nitrogen base, the important thing to remember here, unlike purines, that the sugar molecule is attached to N1 position of pyrimidine ring, that is the N1, that is the position 1 of nitrogen atom in 6-member ring. So when the sugar molecule is attached to thymine, it becomes thymidine, and that is its nucleoside. Now when the sugar molecule is attached to cytosine, it becomes cytidine. Now when the sugar molecule is attached to uracil, it becomes uridine. And lastly, when the phosphate group is attached to nucleoside, it becomes nucleotide, and when the phosphate group is attached to thymidine, it becomes thymidylate, and alternatively it is also called as thymidine 5 monophosphate, therefore it is also called as TMP. And in case of cytidine, it becomes citidylate and cytidine 5 monophosphate, that is CMP. And when the phosphate group is attached to uridine, it becomes uridylate, and it is also called as uridine 5 monophosphate, that is UMP. So again, the transformation from nitrogen base to nucleoside to nucleotide is important to remember here. So just a quick revision. So DNA, like we saw, DNA is a double-stranded molecule, whereas RNA is a single-stranded. DNA is deoxyribose sugar. It is deoxyribose sugar because it lacks hydroxyl group on position 2 of carbon atoms, and ribose sugar, which has 2 hydroxyl group. Now DNA is located inside the nucleus, whereas RNA is located outside the nucleus. DNA is important in storing genetic information, whereas RNA is important in transferring genetic information as well as important in protein synthesis. Now DNA and RNA are made up of nucleotides, and these nucleotides contains a phosphate group, a sugar molecule, and a nitrogenous base. And furthermore, this nitrogenous base, there are 2 forms of nitrogenous base. One is purines and pyrimidines. Purines are double ring, and like I said before, purines are shorter name and longer structure. Pyrimidines, longer name and shorter structure, that is 6-member ring, and that means it's a single ring. Purines contain adenine and guanine, whereas pyrimidines contain cytosine, thymine, and uracil. And just to quickly revise, there are different forms of nitrogenous base which we already saw. Adenine, and when the sugar molecule is attached to nitrogenous base, it becomes nucleoside, that is adenosine. And when the phosphate group is attached to nucleoside, it becomes nucleotide, that is adenylate or adenosine monophosphate. So for guanine, when the phosphate sugar molecule is attached, it becomes guanosine, and when the phosphate group is attached to guanosine, it becomes guanylate or guanosine monophosphate. For cytosine, it is cytidine when the sugar molecule is attached, whereas when the phosphate group is attached, it becomes citidylate or cytosine 5 monophosphate. And for thymine, when the sugar molecule is attached, it becomes thymidine, And when the phosphate group is attached to thymidine, it becomes thymidylate or thymidine 5 monophosphate. And lastly, uracil, when the sugar molecule is attached, it becomes uridine, and when the phosphate group is attached, it becomes uridylate or uridine 5 monophosphate. So thank you so much for watching, guys, and please like and subscribe if you learn something from this lesson, and I'll see you in the next lesson. Thank you so much for watching.